So thank you for coming back, and as I promised you, this session will an awardee session, but we will start with the best secondary school students, and this year we would like to give the prize to Dominik Dobos from Szombathely, who gave a very good performance in last year and showed to the teachers and also the foundations his excellence. And I must say, of course, not only the students needs congratulations, but also his secondary school biology teacher, József Baranyai, who had really a big involvement about Dominik's work. So please, Dominik, provide your research, what you have done last year.
Dear audience, a warm welcome to everyone. My name is Dobos Dominik. I am a student at the Semmelweis University of Budapest at the Faculty of Medicine, and I was a high school student at the Általamújai János Secondary School. And the name of our project that I will now present to you is Annual Model of the Human Blood Circulation from idea to interpretation. It's a great honor to speak to you here. Thank you very much for thinking of me in such bad circumstances and for allowing me to give this presentation. This slide shows the content of my presentation. I try to create a structure that is easy to accommodate and as enjoyable as possible. So let's get started. Here you can find a quantitative comparison of the disease groups with the highest mortality rates in Hungary, according to a data of the Hungarian Central Statistical Office, so-called KSH. And here you can find the deaths caused by the blood circulatory diseases on the left side, and the deaths caused by the malignant tumors on the right side of the slide. And deaths caused by diseases of the circulatory system have the greatest impact on the mortality, and the decline in early mortality was primarily the result of a reduction in early death from circulatory system diseases. And I'm convinced that properly informing people about the circulatory system and the risks involved would lead to a measurable change in the mortality rate. In Einstein's words, if you can't explain it simply, you don't really understand it well enough. So man is a mechanical being made up of a multitude of interconnected systems. In order to understand the complicated things, we need to simplify them. From here, we will be able to draw more complicated conclusions and think further. I think mechanical models make it much easier to understand other similar models if this can be done, and why not? So think of ventilators, which have been voiced a lot lately nowadays, or temporary artificial hearts. So the aim of the project was to create a system of a model that demonstrates the human blood circulation and an OSHA software which together display the nature of the circulatory system, the peculiarities of its operation, its structure, and its illnesses. So the model serves as a demonstration to the study material presented by the software. And it is important to know that the model serves as a demonstration for the material presented by the software. So here you can find the structure of the software, you can see charts, switches, data, and so on, and the control unit of the model. So the brain of the model is an Arduino Uno type microcontroller, which is controlled by a unique on-written software written using the National Instruments user interface, LabVIEW 2016. And this processes all the incoming data as well and shows them graphically, as you can see on this slide. And what about the physical model? So two pumps can be found in the model, just as in the human circulatory system. One is for demonstrating how the right part, and the other demonstrates how the left part of the heart works. The arteries and veins are modeled by transparent elastic tubes, which slightly contract or expand under pressure. And two pressure equalizer tanks can also be seen. One is located at the aorta, and its purpose is to imitate the Owen-Castle effect. The other pressure equalizer tank is at the beginning of the larger skin, which is the vena cava inferior, and substituting the skeletal muscle pump. What about the valves? We use two types of valves. With the 12 volts open-close solenoid valves, fluid supply of different areas can be regulated. This 12 volts open-closed solenoid valves can be found on the left side on this picture. And abnormalities such as stroke, pulmonary edema, or arteriosclerosis can be shown thanks to this. And the non-return valves that can be found on the right, which only let fluids flow in one direction, serve the same purpose as the valves in deeper leg veins. On the slide are the diseases that can be simulated using the model and the software. So what is special is that their effect on our cardiovascular parameters can be observed individually or even together. So stroke, atherosclerosis, hypertension, varicose veins, pulmonary fibrosis, or we can weaken the heart muscle. And we have an opportunity to build in other diseases to the software, and we can measure the changes, and we can calculate risk and so on. And what about the laws? I do not wish to discuss the 4 laws of fluidity. This is for a long time. They are even easier to understand individually, but their combined effects can only be examined with complex mathematical equations. However, with the help of our simulation system, we can observe them simply, intelligibly, and interactively. So, for example, the model is able to calculate blood pressure compensation due to a decrease in vascular radius or vascular cross-section due to atherosclerosis, in addition to which the pressure on the wall of the arterioles can be examined as well. It is very important for us to have students learning anatomy and physiology on an intermediate or advanced level. Understand and learn about this complex system in the easiest way. So furthermore, our goal was also to make the instructors work easier with this model and the accompanying auxiliary material. What is innovation in education? What does it look like in the practice? Here you can see the model in its entirety with a QR code in the middle. If we read this, we can access an educational aid that allows the data provided by the model and the software to be interpreted in detail, but very simply. Illustrations, animations, tasks, descriptions with augmented reality, help with comprehension. And what other things? So for example, let's see examples of innovative solutions. Why is it that the heart works periodically, yet the blood flow is continuous? This is due to the Wien-Cassel effect I already mentioned. It can be seen here with the help of a model to periodically pump the liquid. So let's look at the varimeter. And in the period between the two pressure excisions, the flow will be even with the help of the pressure equalizing value. And surprisingly, it does not work otherwise in the human body. So we have designed tanks instead of balloons that are much more accurate, durable, adjustable in position, and can be used to easily change system fluid through them. And what other innovative solutions? So for example, we can change the overall diameter of veins and arteries using valves. So we can open and we can close channels, and we can calculate the risk, and we can simulate the diseases and the physical laws. We can illustrate the vein functions. So for example, the skeletal muscle pump with the pressure equalizer tanks, and we can simulate the vein valves. We can determine and we can react into the stance of the model using a gyrosensor. we have the possibility to simulate microgravity. The microgravity environment is extremely interesting, as ongoing research is underway on this topic as well. It is absolutely certain that in the state of righteousness, the adaptation of the circulatory system to the environment is completely different from that here on Earth. And we can simulate external effects. So, for example, we can reduce blood pressure. So the user has the possibility to simulate the effects of an antihypertensive drug. We are using ACE blocker, antihypertensive drug, and we can simulate the external effects of alcoholism. So, for example, the different effects of alcohol on the blood pressure of men and women can be seen as well. The software and the auxiliary material is available in English, German, and Hungarian. So thanks to the versatility of the project and the virtually unlimited range of Arduino-compatible products and our enthusiasm, the possibilities of further enhancing the system are most limitless. So lastly, it's important to mention that the Arduino community is huge and open, so the users can easily learn how to improve and personalize the system. The model involves a lot of areas of science. If one wants to understand the operation of the system, they will simultaneously gather deep experience in the fields of biology, physics, and programming. So we were motivated by exactly this enormous resource of knowledge. Furthermore, although it proved to be a rewarding challenge to incorporate the material we studied in at least 3 different types of lessons, the greatest motivation for us was to help our fellow students or classmates. And the acknowledgement, the use of the PR was not accidental. Obviously, I didn't work alone. My teammate was Szeged, who is currently an engineering student at the Imperial College of London, with whom we worked continuously since November 2017. On the implementation of this project. Varanai József, our mentor teacher, is the person to whom I owe the most. He's the one we've worked with from the beginning, exchanging ideas and getting all the support we wanted. So they can be seen on the picture on the left when we won 2nd prize in the 27th Art Science and Innovation competition in June 2018. As a result, we were able to get to China, the 34th China Laws and Science and Technology Innovation Contest, which is the 2nd biggest scientific world expo, where we could represent the Hungarian nation. Our team leader was Jakab László, the chairman of the jury of the Hungarian Innovation Competition, a former dean, and the professor of Budapest University of Technology and Economics. In Macau, in China, we won the biomedical section, and in total, we became 2nd grand prize winners, silver medalists. In addition to them, many others stood by us, such as Szabó Bence Farkas, to whom we can also owe a lot. And last but not least, I would like to say a few words about Szeged Scientists Academy. As part of the program, I was given plenty of opportunities for development, be it lab exercises or panel discussions. At similar conferences, I was able to make lifelong friendships, meet Nobel-laureate scientists. The whole atmosphere inspired me every time. As a high school graduate, I can say for sure that there is no better opportunity for development with this program if one wants to engage in science research. Take advantage of your opportunities. I think they will help you in everything here. Despite the fact that I am not studying in Szeged now, seeing the circumstances and the determination and the faith in science, I think it is quite likely that I will continue my scientific career here and live with my family here in Szeged in the future. Here are my references that I used to do this presentation. And in conclusion, to be able to speak to you here now, it is due to 3 things. The mentors and peers who have always stood by me, The second thing is humility. And the third, most important thing, the previous two must never be forgotten. Thank you for your attention.
Thank you very much, Dominic. When I heard your presentation, I wouldn't be able to say whether you are a school-living university students or a school-living secondary students. This research was really a very high level. So congratulations again to you and also your teacher. So let's go further. And now the University Excellence Award will be given. And this year, Valéria Éva Meszlényi will get this work. And her junior mentor was Roland Patai, and the senior mentor László Siklós. Congratulations to all of you, but of course we would like to get the presentation from Valéria Éva Meszlényi. Please provide your research summary to the audience.
Dear Professor, thank you very much for this award. It is a real honor. Ladies and gentlemen, dear students, I am very grateful for this opportunity to give a presentation here today. However, it is not an ordinary conference because all of the presentations are online due to the current state of the pandemic. I think that this situation draws your attention and maybe diverts attention to the importance of biomedical research, because nowadays everyone's interested in coronavirus, everyone's interested in the arrival of the vaccine. However, to learn about these phenomena, we need research. And it is not only true for coronavirus. The pathomechanism underlying a number of diseases is still unknown or not fully discovered, and patients and doctors all over the world are waiting for a breakthrough which can lead to novel therapeutic approaches. This is also true in the case of amyotrophic lateral sclerosis or ALS, because there are a lot of things regarding to the pathomechanism of ALS, what we still don't know, and there is no cure for ALS yet. Our research group in the Biological Research Center in Szeged is mostly interested in amyotrophic lateral sclerosis. And in the first part of my talk, I would like to tell you the basic facts about ALS. Then I would like to present our current research. ALS is the most common motor neuron disease, and as its name says, motor neurons are affected. That means that these neurons in the brain and spinal cord degenerate, and as a consequence, the message from the brain stops reaching the muscles, and it leads to weakness and paralysis. If we think about it, we need muscles for almost every activity, like to speak, to walk, to drink, to eat, and to breathe. And in motor neuron disease, these could be all affected. ALS can be classified as familial or sporadic, whether there is a family history of the disease. Degeneration of upper motor neurons in the brain cortex and lower motor neurons in the brainstem and spinal cord leads to weakness and paralysis. Because of the involvement of respiratory muscles, patients usually die within 3 to 5 years through respiratory failure. The diagnosis of ALS is difficult, and as I mentioned, there is no cure for ALS yet. I wonder if you remember the Ice Bucket Challenge back from 2014. The goal of this challenge was to promote awareness for ALS and to encourage donations to ALS research. Famous people like Bill Gates in this video and Mark Zuckerberg also participated in it. There are plenty of processes contributing to motor neuronal degeneration in ALS, such as oxidative stress, glutamate excitotoxicity, mitochondrial dysfunction, elevated intracellular calcium level, and I would like to highlight the autoimmune and inflammatory processes. Our research group previously proved that in the blood serum of ALS patients, there are anti-motor neuronal antibodies. These antibodies can alter with calcium channels in motor neurons and can contribute to increased intracellular calcium level, which leads to degeneration. In our current experiment, we used the blood serum of ALS patients with identified mutations. The background of this present study is that our research group previously proved that in the muscle biopsy samples containing motor axon terminals of sporadic ALS patients, there is an increase in intracellular calcium level and increase in the number of synaptic vesicles. Later we proved that these alterations could be conferred to mice via the passive transfer of the sera from these sporadic ALS patients. After that, we proved that similar alterations can be found in the transgenic mouse model of familial ALS in the SOD1 transgenic mouse model. These observations suggested that calcium may play an important role in the pathomechanism of both sporadic and familial ALS. This could be further reinforced by completing a similar analytical study as in the case of sporadic ALS patients. However, such an experiment is not technically feasible because of the low yield of muscle biopsy samples containing motor axon terminals and the low incidence of the disease. Alternatively, in our current experiment, we used the passive transfer and we performed intraperitoneal injections with the sera from patients with identified mutations, and we investigated the motor axon terminals of mice after the passive transfer to see whether there are similar alterations as in the case of sporadic ALS model and transgenic mouse model of familial ALS. To do that, we performed passive transfer for 2 days with the sera of patients with ALS with identified mutations. After that, we performed transcardial perfusion and fixation, and the interosseous muscles were removed, and the motor axon terminals found in the interosseous muscles were analyzed under electron microscope to detect intracellular calcium and synaptic vesicles. In this slide, I would like to show you some electron micrographs of motor axon terminals. At the left, we can observe 2 healthy synapses, one from the untreated control group and one from the healthy serum-treated group. We can observe that there are only a few electron-dense deposits, which represents calcium, and mitochondria are well structured. If we take a look at the right, there are synapses from the ALS serum-treated groups, and if we compare this with the control groups, we can observe that there is an elevation in electron-dense deposits compared to the control groups, and the mitochondria are swollen and damaged. In the following slide, I would like to show you a number of synaptic vesicles. These synaptic vesicles, or neurotransmitter containing vesicles, are essential in the propagation of nerve impulse between the neuron and the muscle. So in these images, we can observe in the control groups only a number of synaptic vesicles in the active zone of the motor axon terminal. But in the case of ALS serum treatment, there is an elevation in the number of synaptic vesicles in the active zones of synapses. And here we can see our results. After we quantified intracellular calcium with geometrical statistics, we observed that after the passive transfer with the sera of ALS patients with identified mutations, we can detect an increase in intracellular calcium level compared to the untreated and healthy serum treated control, as well as in the case of sporadic ALS sera. If we take a better look, we can observe that in the case of C9orf72 mutation, the calcium increase was significantly higher compared to all other ALS sera as well. We quantified the number of synaptic vesicles as well, and though we proved that due to the passive transfer of ALS sera with identified mutation, there is an increase in the number of synaptic vesicles compared to the control groups, as well as in the case of sporadic serum treatment. We detected significant increase in both of the examined parameters, and because they are closely associated with the activity of synapses, their cross-relation was analyzed with cluster analysis. In this figure, each point represents a patient with a mutation, and we can observe three main clusters: one for the controls, one for ALS patients, and one for ALS outliers, representing the patients with C9orf72 mutation. However, it was not our goal to analyze the difference between the identified mutations. It is noteworthy to mention that the effect of the sera from the patients with C9orf72 mutation exceeds the effect of all other ALS sera. This might be because of the strong inflammatory effect of this mutation. Now I would like to sum up my talk. Due to the passive transfer with the sera of ALS patients with identified mutations, we observed an increase in intracellular calcium level and an increase in the number of synaptic vesicles, as well as the passive transfer of the sera from sporadic ALS patients. So our results support the idea that the pathomechanism underlying the identical clinical manifestation of both familial and sporadic ALS is based on a common final pathway in which calcium plays an important role. Moreover, C9orf72 mutation were sorted into a different cluster with cluster analysis. At the end of my talk, I would like to show an overview of the pathological processes of ALS. As I mentioned in the beginning of my presentation, The main elements of the pathomechanism of ALS are oxidative stress, mitochondrial dysfunction, inflammatory and immune processes, and elevated intracellular calcium level. Let's see in details how they operate in an orchestrated way and amplify the effect of each other. Here we can see an excitatory axon terminal, a motor neuron, and a skeletal muscle. Increased intracellular calcium in the excitatory axon terminal leads to excessive glutamate release. Glutamate is the most common excitatory neurotransmitter in the nervous system, and it binds to glutamate receptors. AMPA receptors are a subtype of glutamate receptors which are permeable to calcium in ALS due to a conformation change, which leads to elevated intracellular calcium level in motor neurons. The elevated intracellular calcium in motor neurons has a lot of consequences, such as the activation of endonucleases and proteases, which actually cause DNA damage and the destruction of cytoskeleton, which is a protein filament network. Another consequence is the activation of enzymes which produce reactive oxygen species, which lead to membrane injury. Mitochondria are also impaired due to the elevation of intracellular calcium, which leads to energy deficit and the formation of even more reactive oxygen species. As a result of these pathological processes, neurons release stress signals that activate the neighboring glial cells, microglia and astrocytes, which produce even more reactive oxygen species. The distal part of the neuron is also damaged because of the elevated intracellular calcium level in the pericardia or because of the anti-motor neuronal antibodies. These mechanisms have strong and reciprocal calcium dependence, and calcium forms the individual pathological processes into a unified escalating mechanism of neuronal degeneration. To conclude, calcium not only plays a role in the certain subtypes of ALS, but regardless the cause, it is universal in the pathomechanism of ALS. Now I would like to thank my supervisor, Dr. László Siklós, for his help and guidance, thank my junior mentor, Dr. Roland Patai, for his support, and thank the members of our research group and of course our collaborators. And now I would like to thank you for your attention.
So it is my pleasure to announce the this year 2020 Talentum Award for László Nagy, who works in the Biological Research Institute of Szeged in the Department of Biochemistry, and the award is for his contribution in the fungal genomics. And I would like to congratulate to your very prestigious and valuable contribution.
Thank you very much.
I also would like to hand over to you.
Thank you so much.
Thank you.
I appreciate. It's really an honor to be awarded this prize. I really appreciate all the participants and all the organizers of this foundation. I feel really honored to be among the awardees of this foundation. Thank you very much. Greetings everyone. It's a real honor to be here. I really appreciate the nomination and the award of the foundation. Thank you very much. Today I would like to talk about fungal genomics and how genomics and evolution can be combined to understand some of the major questions and challenges in biology and evolution in particular. So genomics as a field is relatively young. It was born when the first genome sequence was completed, and soon after, when the second genome sequence was completed, comparative genomics was also born as a field. So as you may all know, the genetic code that is encoded in our DNA can be read out, it can be sequenced, and this gives us a infinitely long code of 4 letters that code for the basis of the DNA. This long code is a pretty boring read if we wanted to read it as a book, but it contains the code for life and the code for the species-specific traits and behaviors of organisms. And if we have a key to understand this code, then we can also compare organisms and learn about differences and the genetic determinants that dictate how organisms develop, evolve, and live their lives. So one of the first genomes, or some of the first genomes, were prokaryotic and eukaryotic microbes. These belong to model organisms like Escherichia coli or Saccharomyces cerevisiae. But soon after, the Human Genome Project has also started. This was a humongous undertaking which took some 15 years to complete, and it costed around $2.7 billion at the time. So it's a really huge project that basically resulted in cracking the DNA code of the human species. As I mentioned, this code can be represented as a plain text, and if we were to put this text in a book format, that would require 130 thick volumes of books in a printed format. So that's a really huge amount of information. Now, recently, with the advances of technology, new sequencing approaches have evolved, and that really gave a boost to genome sequencing projects. I mentioned that the Human Genome Project took around 15 years. Today, sequencing a new genome really takes less than a few days if we only talk about the sequencing part of the project, and it takes an additional several days or even months to actually analyze those genomes. Now that we have really fast and cost-effective sequencing technologies, we can really aim for sequencing the genomes of tens, hundreds, or thousands of species. And that gives us the opportunity to answer questions about evolution, to answer questions about disease and the behavior or the morphological development of various species. As a response to this availability of technology, a large number of really large-scale genome sequencing projects have been started. One of these, the 1,000 Fungal Genomes Project, which lies closest to my heart, was started in 2015. It was one of the first of its kind, but there are a large number of other sequencing projects underway as well for various organismal groups, insects, birds, vertebrates, and even some projects like the Earth Biogenome Project are aiming to sequence all living organisms on Earth. So this is a really ambitious undertaking, and we can see that these projects will generate huge amounts of data. And one of the questions we have to ask is how to analyze all this data. This is where bioinformatics comes into the picture, and while I mentioned before that for the Human Genome Project, sequencing and laboratory work represented the bottleneck, today this is not anymore the case. Today, bioinformatics represents the rate-limiting step in scientific discovery and research and publication, which means sequencing is really fast, bioinformatics can be much slower. It requires really highly skilled bioinformaticians. high-performance computing architectures, and so on. So if I was to give an advice to the younger generation here, I would really encourage everyone to study bioinformatics and become a bioinformatician, because it's one of the most highly sought after specializations in biology today. So the general logic of comparative genomics is that once we have multiple genomes available, we can ask questions about what is common and what is different in those genomes. Let's assume we have a group of species that possesses one trait, and we have a group of species that doesn't. And then we compare these two groups, and in the ideal case, we are able to answer what are the genes or genetic elements that underlie the difference between these two species. For the example here, I'm starting with a pairwise comparison of species. Let's assume that we have two genomes at first, which belong to species 1 and species 2, and species 1 has a blue phenotype, and species 2 has a yellow phenotype. We are interested in what are the genes that cause the difference in color between these two organisms. So if we analyze these genomes, we will see a large number of differences between the genome sequences. Really think about hundreds of thousands, if not millions, of differences between any two genomes that we can compare. We don't really have a mean to distinguish between important and non-important differences. So the solution of comparative genomics to this problem is that when we add more species to the system, we can achieve a higher precision in assigning differences between species and between genomes to actual traits and observed phenotypic differences. So for the example, I added 4 more species to this data set, and I also drew the phylogenetic tree that connects these species with each other. You can see that 3 species have the blue phenotype, and another 3 species have a yellow phenotype. And if you look at the phylogenetic tree, it's also obvious that the changes between the differences between species can now be located to specific branches of this phylogenetic tree. So that allows us to compare these differences between the evolution of the phenotype that we are interested in. So we can use again bioinformatic methods like ancestral character state reconstructions to infer where on this phylogenetic tree the trait has changed. And if we do so on this example phylogeny, we would find that the yellow phenotype was the ancestral condition, and then there was a change, possibly a mutation, some innovation that resulted in the blue phenotype. And once we know where this change happened in the trait, we can, on the other side of this slide, focus on the same branch, on the exact same branch on the phylogenetic tree, where we would expect the really relevant differences between 2 species to occur. So this is how comparative genomics can really help us understand species differences and the underlying genetics. Now in my laboratory, we are interested in fungal evolution and fungal multicellularity. For one, because multicellularity has been one of the greatest innovations in the history of life. We ourselves are multicellular organisms. Most of the organisms that surround us, trees, mushrooms, all kinds of algae, or most kinds of algae, are multicellular animals, or multicellular organisms, excuse me. So it has been a huge question for biologists. What are the genetic determinants of the evolution of multicellularity? In the case of fungi, multicellularity is realized by apically growing hyphae. You can see on the video that these hyphae are growing really fast at their tips, and they invade new environments and new habitats with this fast apical growth. They do this they branch in a fractal-like manner, so that the branching events are filling the available space in the most optimal manner. This might enable them to avoid conflicts between individual hyphae, which is often a great problem when multiple individuals come together and they have to cooperate with each other to be able to achieve a higher fitness. On the lower right video, you can see how a mushroom develops. A mushroom is a complex multicellular structure that develops from fungal hyphae. And this is a really complex and really well integrated developmental process that needs to be orchestrated very well in time and space, so that the exact species-specific morphology arises. So we were thinking, where is the advantage of fungal multicellularity? And one of the fields where fungi really excel is terrestrial habitats. When food sources are patchy, these long filaments, the hyphae, can really efficiently bridge over nutrient-poor areas, so that they can connect areas where there is one type of nutrient and where there is another type of nutrient. And so this transport system that they can set up in the soil or in the substrate really enables them to colonize a large number of terrestrial habitats. It also enables them to degrade really complex biological materials. One of these complex materials is wood. So wood is produced by plants, of course. It's one of the most abundant organic polymer on Earth that forms really resistant structures. Just think about how long a wooden structure can last in history. It's really hard for microbes to degrade woody materials. And if we look around on Earth, we see that actually there are over 2,000 gigatons of organic carbon sequestered in various woody structures. So this is a really huge sink of carbon that contributes to the carbon cycle and can contribute also to sustainable industries if we can smartly utilize it. And fungi are able to do just that using their extracellular enzymes, which are secreted from the hyphae to the environment. I would like to mention a very interesting hypothesis here that we worked on back in 2012 and before. It looks like from molecular clock analysis and comparative genomic analysis that fungi might have something to do with the end of the Carboniferous period. So this phylogenetic tree on the right-hand side of the slide shows the relationships between 33 genomes of wood-decaying fungi, and this is also time-calibrated. So when we project the place when really efficient wood-degrading fungi first evolved on Earth, then we see that this coincides with the end of the Carboniferous period, around 290 million years ago. So the hypothesis comes that maybe these really efficient wood-degrading fungi were the first players who could decompose wood that was otherwise, during the Carboniferous period, accumulating in the soil and buried under the soil, and gave rise to fossil fuels, coal deposits, and so on, that we exploit today for transportation, for industry, and so on. So when we are about to blame something for running out of organic or running out of coal and mineral oil, then we might even blame fungi for it. These enzymes that fungi use to digest wood can also be used in the biofuel industry. So the biofuel industry is based basically on solar energy, with which plants fixed carbon, they grow biomass, and this biomass is used in various biofuel factories to produce simple few carbon molecules that can be used in vehicles. Now, there is a really hard part of biofuel production, and that is decomposing the cellulosic plant material into much simpler components. And this is where fungi can be really efficient. The fungal enzymes that evolved during hundreds of millions of years to decompose wood can be used, if we are able to identify them, they can be used in the industry to facilitate the production of biofuels. And this is one of the major aims of the 1,000 Fungal Genomes project, in which we are also involved with our Szeged laboratory. And this was the aim of some of the work we are doing in my laboratory. So in a previous study, we compared around 70 genomes of wood-degrading fungi, and we used computational methods to predict the gene families and the enzymes that can be really efficient in wood decomposition. And we did find around 300 gene families, about half of which was not known to be involved in wood decay before. So I think this illustrates really well that computational approaches and bioinformatics can be really efficient for finding new players in the biofuel industry. It should be mentioned that all this work requires large supercomputer infrastructures, such as the Cori supercomputer, on which Balázs Bálint from the laboratory is working a lot on. He is the master, I would say, of supercomputing infrastructure in my laboratory, and maybe also in Szeged. Now I would like to loop back to fungal multicellularity after this small detour into extracellular enzymes. So multicellularity has been a major transition, and because it's a major transition, biologists have envisioned that it coincides with a large amount of genetic innovations. This we can imagine as a large number of new gene families, gene duplications, or other types of innovations in the genomes of multicellular fungi that happened at the time when unicellular fungi became multicellular. So for that we can use a comparative genomics approach, and we can ask what are the systematic differences between multicellular and unicellular fungi. For that, a PhD student in my laboratory, Enikő Kis, started to dig in the literature, because we know hardly anything about fungal multicellularity, and especially the origins of it. But fortunately, we know a lot about the genetics of hypergrowth and its regulation. So Enikő went to the literature. She searched papers for genes which are known to be involved in hypergrowth, and then examined the evolution of these genes. For that, she assembled a whole genome dataset of approximately 80 species, and first performed an ancestral character state reconstruction, like the one I showed you in the beginning. And this ancestral state reconstruction suggested that the first multicellular fungi evolved at the base of the fungi, around the place where you can see the black square on the slide. So we know now that this is the place where we should really focus our searching efforts for finding genetic innovations that might explain the evolution of multicellularity. And so Enikő went on and she reconstructed the evolution of these genes, and what she found was really surprising. We expected that a large number of genetic innovations would be happening at the time when fungal multicellularity evolved, but in fact what we see is the opposite. And that was a very interesting observation. We saw that there are very few new gene families and gene duplications that happened in the nodes where multicellular fungi first evolved. And on the other hand, there are a lot of innovations before and after. So if we were to summarize this lack of a major burst of an evolutionary innovation in a pie chart, then we could say that new gene families and gene duplications are less influential than we thought before, and processes so-called co-option and exaptation are much more common. So co-option is the process when an existing gene that has some kind of function in the ancestral species is recruited for a new functionality, in this case multicellularity, and afterwards it serves the function of multicellular organisms. So it looks like in fungi this is the dominant mechanism, and this resembles the blind watchmaker metaphor of evolution, which says that evolution tinkers, it does small random changes here and there. Those are not purposeful changes. There is no purpose at all in those, but it works with what is available. It's like, you can think of it as really a watchmaker that has a workshop around, and whatever part is available, he or she would incorporate that into the watch or into the innovation that he or she is working on. With this metaphor, I would like to sum up my talk. I hope you can remember and take this as a take-home message with you, and thank you for the attention very much. Thank you.
So thank you very much for being with us. So now there will be a break, and please come back at 4:30.